Hello and welcome back to lecture 9.2, where we will be finishing off the cellular respiration process, including the steps of the citric acid cycle and oxidative phosphorylation. Now, before we get to the content for lecture 9.2, let's do lecture question 9.1. Compare and contrast the products of glycolysis and pyruvate oxidation. Neato. If you said A, you are correct. If you didn't say A, no big deal. Just go ahead and review last lecture. Maybe try to make an organizer comparing and contrasting glycolysis and pyruvate, like a table perhaps. You could also maybe, I feel like there's something out there that helps you look at products. Maybe it could help you compare and contrast the processes of glycolysis and pyruvate oxidation and the products that come out of those processes. Anyway, moving on. Lecture 9.2, Cellular Respiration, Part 2. Okay. So, we're going to start with the next stage in this process, the citric acid cycle. The citric acid cycle takes place inside the mitochondrial matrix down here, which is basically just kind of this extra dense cytosol inside the mitochondria, inside the folds of the mitochondria. So why did we do this? Well, again, the idea is to kind of break down that acetyl-CoA as efficiently as possible and build those electron acceptors that we're going to hand over to the electron transport chain in just a moment. Now, don't forget, this is for breaking down acetyl-CoA. And how many acetyl-CoA do we have per molecule of glucose? Right, 2. So the citric acid cycle needs to happen twice for each glucose molecule. And remember, this is basically a series of a ton of kind of reactions here that we're going to kind of glaze over the details of all these intermediate steps. Maybe in a more advanced course you'll go over it, but we're going to focus on kind of the big picture here so don't get too crazy about it. Okay. Now, first step of this process is we have a kind of 4-carbon molecule called oxyl acetate that's kind of just hanging around in the matrix waiting for the acetyl-CoA with its 2-carbon acetyl group to show up. Now, when it does, enzymes will actually join the 2-carbon acetyl to the 4-carbon oxyl acetate, forming a 6-carbon citric acid. So what do we think happens to that coenzyme A that's been moving this acetyl group along? It actually gets reused. That's handy. Now, we then have a bunch of reactions that start pulling that citric acid apart, handing off the electrons to various electron acceptors, including NAD+ and FAD. Now, what happens to the NAD pluses and FADs when they've grabbed an electron? Right, they get reduced, reduction is gain, to a bunch of NADHs and FADH2s that are going to get used later on. Now, while these different electrons are getting pulled off, we're also oxidizing the remaining carbons from that glucose molecule that are left over, right? Those 2 carbons that were in the form of the acetyl. So we're actually going to be pulling off more carbon dioxides, and this ultimately results in that glucose molecule being completely oxidized and 2 more molecules of carbon dioxide are produced. You also generate 1 little ATP from this whole process. Pretty straightforward here. Okay, now let's simplify this down and maybe check our math a little bit. You have one turn of the citric acid cycle per acetyl-CoA, right? Which acetyl is the leftover carbon. CoA is the coenzyme that kind of helps shepherd this acetyl group along. So you have 2 acetyl-CoAs enter the cycle per glucose molecule. It takes 2 turns to break those down and generate 4 carbon dioxides, 2 ATP, 6 NADHs, and 2 FADH2s. So we have our running tally up here, and so far this all checks out. We've actually gotten rid of all of our carbons from our 6 carbon molecule of glucose. We've produced, we lost a couple in the beginning to the investment phase of glycolysis that we gained back, and then we gained a little bit more ATPs. Now for NADHs, we've just been pumping them out. We had a couple initially that we made in glycolysis and then pyruvate oxidation, and then we had a bunch made just now. And then of course we also had a couple of FADH2s made just now as well. So what are we doing with all these FADH2s and NADHs we've been putting together at this point? I'm going to use them during this grand finale here, the oxidative phosphorylation process.